Welcome, everybody. Thank you for coming. So today, it's a real honor for me to be able to introduce Stephania Cormier. So a little bit of background on Steph. She got her bachelor's degree from the University of Southwestern Louisiana. You're going to hear a trend here. And her PhD also from LSU. Originally, she worked on developmental biology and cardiomyocytes. And then for her postdoc, she switched towards immune signaling and eosinophil function. She then got a faculty position at LSU. And then she moved to the University of Tennessee at Memphis, which is where we first touched base. And then she has since moved back to LSU. So she's a big fan of Louisiana. She's here to tell us about how to make our football team a national championship contender, amongst other things. So Steph is currently, there's a lot of this listed here. She's the AVP for research at LSU. She's the director of their LSU Superfund research program, which is on combustion-derived particulate matter. And funded by P42 from NIEHS. She's a professor of Comparative Biomedical Sciences and a Professor Wiener Chair and Professor of Biological Sciences. So her research, she's going to tell us only about one small part of her research, but her research is really into a lot of early life exposures to viruses, infectious agents, and environmental pollutants. And she really does a really astonishing job of basic science combined with epidemiology, combined with translational science. She really covers the gamut of all research. And then for me, this is a big thrill. Steph is one of my primary mentors. She's always been a big advocate for me. She's to blame for all of my faults, or most of my failings anyways. But she's been a real inspiration for how to take a basic science program and really think about how to translate it to public health, which is, I sure would not be working in public health if it wasn't for that perspective. So Steph is currently funded by a P42 and 2 R01s. And thanks to M-LEEaD for sponsoring today's visit. So, all right, and so Steph's going to talk about maybe e-cigarettes and implications for childhood obesity.
Thank you, David. I haven't even started yet. Anyway, thank you everyone for inviting me to the University of Michigan in January. I'm from Louisiana. I'm a native Louisianian, so that weather out there is like, should be inside cooking a gumbo. But it's beautiful. I've enjoyed the snow so far. It's nice to kind of see it fluttering outside. I appreciate everybody for inviting me and those who have sponsored my trip here. It's been a pleasure so far meeting everyone and discussing their research projects, and I hope that we can continue these conversations maybe in future collaborations. I've already collaborated with some people here, and I hope to bring back more collaborations. So today I'm going to talk to you about one of my projects, as David mentioned. I do quite a few things all around infancy and trying to figure out what during infancy leads to long-term airways dysfunction. So this is a small portion of this. We were actually looking at in utero exposure to e-cigarettes as a joint project between me and David that we're currently working on. Okay. All right, so a lot of my work, an R01 and the Superfund project, is actually looking at combustion or thermal-derived particulate matter. And combustion-derived particulate matter comes from a variety of sources. Most of those sources we know. There are mobile sources such as diesel exhaust engines. There are stationary sources, power plants, factories, even fireplaces. There are natural sources like forest fires, bushfires like in Australia, and volcanoes. And then there are personal sources such as cigarettes or e-cigarettes. Now, this was the only graphic that is weird, so I apologize, and I hope you can see through this. But the bottom line is that if you take combustion-derived particulate matter, and this is where my studies in environmental tox actually started. I had a chemist, Dr. Barry Dellinger, who's no longer with us, approach me when I was an assistant professor at LSU, and he was interested in understanding how particular components of particulate matter were involved in the health effects associated with particulate matter. He was a chemist. So he had no clue how to study it, and so we sort of stepped in to help him. And his idea was that the presence of these radicals, which we now call environmentally persistent free radicals, which I'll tell you about later, is present in pretty much any soot-type complex, unless you're even from incinerators, but mostly they're formed during poor incineration or thermal remediation processes. And his idea was that, okay, I can't even show it. Wait, I gotta do this. Okay. Okay, so I'm going to show you here. So his idea was that these radical species that are present in soots from a variety of incineration processes are also present in things such as diesel exhaust particles, gasoline exhaust particles, wood smoke, and cigarette smoke tar. So the presence of these radicals are endemic in anything that's actually being burned. And so we actually eventually went on to look at e-cigarette particulate matter. And so this is an electroparamagnetic resonance spectrum showing that these radicals are also present in e-cigarettes. Now, I'm going to switch back to this just because it's bigger. And now it's there. Okay. Now, the level of these radicals depends on what you're actually looking at. So down at the bottom, you can see that cigarette smoke tar has 1 times 10 to the 16th radicals per gram of particulate matter. E-cigarettes, total particulate matter has 2.6 times 10 to the 15th radicals per gram. So almost a log less, but it's still present. But it's present at a pretty high value. And then depending on where you take a PM2.5 sample from, which depends on what they're burning in that area, or the traffic in that area, or the industry in that area, radicals can range anywhere from 1 times 10 to the 16th all the way to 1 times 10 to the 19th radicals per gram. This is Baton Rouge, 1 times 10 to the 16th to 17th, and then this is Memphis, 1 times 10 to the 17th to 1 times 10 to the 19th. And although I'm not going to talk about it now, what we can do is actually geographically map high incidences of pneumonia in pediatric patients, and they map to areas where where EPFR levels are high in the 1 times 10 to the 19th range, but PM levels are actually low. Okay? So it's not just the PM that's the problem. So why do we call these things EPFRs? So we call them environmentally persistent free radicals because I had an experiment, and I was like, okay, what's the control for this experiment? I'm going to take environmentally persistent free radicals, and I'm going to expose cells or animals to them. But how do I control to see if it's the radical that's causing that response? So in my biological thinking, I was thinking, okay, I'm just going to sit this out on the bench until it decays. Okay? So I did that. The problem is it took 21 days of a half-life to decay, and at 54 days, you could still measure radicals present on those particles. Now, this is Baton Rouge PM2.5, but if you gather PM2.5 from different areas, so for example, we have Australian samples where the radicals are decaying over years. Okay? So these radicals are small, they're attached to a particle, and they're capable of going into the lungs. And they're capable of getting, because of their small size, to the lower airways. Okay? So the majority of PM2.5 is like 70% of ultrafine particles. And then this is how they're formed. Now, I'm not the combustion chemist, so this is work done by Barry Dellinger and Slavo Lomnicki, who are combustion chemists. And what they've shown is that in low or inefficient thermal processes, so temperatures of about 300 to 150 degrees C, what happens is aromatic compounds chemisorb through a metal oxide to a surface. And this is a chemisorb species, meaning that it doesn't break apart readily. You either need a strongly acidic or strongly basic environment to dissociate these into a metal, an organic, and a particle, or you need a solvent extraction. So what does the EPA actually do, or most people actually do when they collect PM samples? They dissociate it into its individual components to try and figure out which individual component is actually causing the toxic effect. And by doing that, they actually lose the radical species. So our idea with PM was that this radical species was actually, as a complex, was responsible for the health effects associated with inhalation. Now, what's really even more interesting about this is, so remember we sat it on the bench for the control? Well, then we went back and put it on cells. And so we had our radicals, and we had the radicals we sat on the bench, which should have had no radicals. And we looked for oxidative stress. Both of those samples gave us oxidative stress. Now, the value of the Superfund research program is you call the chemists up and go, what the heck is going on? And so Barry said, we were wondering if this might happen. I'm like, okay, what's happening? And he said, well, we thought that these would redox cycle in a biological system. And because they redox cycled, they would form superoxide and hydroxyl radicals. And in fact, they do. So once you put these particles inside, not only are the particles carrying free radicals, but the particles are initiating the production of free radicals. And in fact, about 10 hydroxyl radicals per particle, depending on the components of that particle. And I'll tell you a little bit about that in a second. And in biological systems, these radical species continue to persist. So this is actually BAL fluid, and this is the presence of these radicals in bronchoalveolar lavage fluid. And you can see that the half-life is somewhere around, I don't know, 5 hours, but they persist all the way through 12 hours in biological fluids. And this entire time, they're making hydroxyl radicals. So we've done some other studies, and those other studies have indicated that the radical content and the lifetime of the radical content, first of all, the lifetime of the radical content is dependent on the metal oxide that's present in that complex, except for zinc, which we're trying to figure out. But zinc is probably the most stable species. And this correlates with a lot of epidemiological data on PM2.5, which correlates zinc to larger incidences of respiratory or cardiovascular effects. In addition to correlating with the lifetime of the metal, the amount of metals actually also affect the production of EPFRs. So depending on how much EPFRs are actually present in the complex, you have more or less EPFRs that are actually formed. Okay? And then this also affects the amount of dioxin U. And depending on the types of metals, and even complex metals, it's a little bit harder to study. So on this axis, you actually have iron and copper mixes. Okay? And so depending on the mixes of iron and copper, which are present in most combustion processes, you can have more or less dioxin formation. So metal oxide content determines the lifetime. The amount determines the amount of EPFRs and also the amount of production of dioxins. And so we've been studying these things, and we were interested in the immune response if we expose pregnant moms to these things. So we have a whole other paper that I'm not going to discuss here, demonstrating that if you expose pregnant mice, to these environmentally persistent free radicals, which are found in e-cigarettes, then what could happen is those moms became Th2 predisposed. So they were predisposed to develop allergies and asthma. And that effect lasted for 6 weeks post-birth, even without a challenge. Now, at 6 weeks post-birth, they came back down to baseline. So Th2 and Th1 responses in the lungs of those animals were no different than controls. However, if you allergen challenged them or SB infected them, then their Th2 or allergy or immune responses were actually exacerbated, indicating that we were changing something in the pups from the in utero exposure or the maternal exposure of mom. Now, while we're working on this project, my postdoc, Sri Jaligama, who currently works at Battelle, was walking down the hall in pediatrics at Labana. And I was walking the other way. And he goes, "You know, Dr. Cormier, the pups from those mice look bigger." And I said, "Sri, you're a postdoc. What does bigger mean? I mean, are they heavier? Or are they rounder? Or are they longer? Go give me some data." And so he came back and they were heavier. I said, "Wait, I know a person." And I contacted David. And so I saw him walking down the hall. And so we went to David and we asked whether or not this could be responsible for increased obesity. And we asked this because of data that was starting to come out. So this is particulate matter levels in the United States. This is PM2.5, satellite-derived PM2.5, which is actually a pretty accurate rendition of PM2.5. Now, this is averaged from 2001 to 2006, so it's not an annual. But what you can see from this is that PM2.5 levels are high here, in this area here, and then also on the West Coast, right? Hi. And then, and the annual PM2.5 levels are supposed to be here, and the daily means are supposed to be maximum of 35. So this just gives you an idea of how high these levels are, okay? And then you look at obesity in children. Notice the areas of high obesity in children here and here. It's the same sort of areas, right? So the question was whether or not exposure to EPFRs, which are things that are present in not only PM2.5 but also e-cigarettes, could lead to obesity. And so to do that, we also looked at other data, I'm sorry. So we also looked at human data, because most people, when you talk to them about EPFRs and or e-cigarette or cigarette exposure, they said, well, most people who are exposed to cigarette smoke exposure in utero actually are underweight. And so this is actually a group of studies that argue against that. So you have several different, you have case cross-sectional studies, you have prospective studies and retrospective studies. And you can see that the majority of these all show an increased relative risk of either overweight, which is in red, or obese with maternal smoking. And while the mouse data is not as clear, but in general the same thing is seen. So here we have nicotine, we have cigarette smoke, and we have nicotine again, and we're looking at body weight or adiposity. And what we show is that prenatal treatment with either nicotine or cigarette smoke leads to slight increases in prenatal treatment lead to slight increases in weight or adiposity, and or prenatal treatment and lactation treatment lead to increases in adiposity and body weight. And this is some of the work we were going to follow up with David, using real e-cigarettes and exposures in animals. And so when we went to David with this data with EPFRs, we had exposures going on during gestation. These exposures occurred during what are the second and third trimesters of gestation in the mouse. We let the mice grow up, so they had 10 weeks post-birth on a regular diet, nursing a regular diet, and then we exposed them to high-fat diets, where 45% of the calories were from lard, and they stayed on that diet for about 12 weeks. And all of this was done in collaboration with David, and all of the obesity stuff was David, and we did this, okay? And his lab, and him. And so this is what we found, and so this was what was intriguing. So again, we saw the same result Sri stopped me in the hall to talk about. So here, MCP 230 is actually one of our model environmentally persistent free radicals, and then we had saline. We have other controls that you'll see as we get through some of these slides. But the bottom line is that animals that were born to moms who were exposed to EPFRs actually had increased weight compared to animals that were exposed to air in gestation, and that increased rate continued throughout life with about 10% heavier body weight at 12 weeks on the diet. Now, this was due to increases in fat mass, and it was due to increases in fat-free mass, so they were just larger, right? Now, and I have to say, these were males only at the time. So we questioned whether or not they're actually eating more, right? So if they're eating more, then that could explain why they're bigger. And so David and his lab actually went in and looked at caloric intake per week and then cumulative caloric intake. And what they found is that, and these are done at a cage mother aggregate level, correct? Right. And what they found is that animals that were born to moms who were exposed in utero, to moms that were exposed to EPFRs, actually took in less food, and their cumulative food intake was dramatically decreased or significantly decreased. So they weren't eating more. So the question is, why? So one of the things we looked at were hunger hormones. And in these infants, so we have two different groups. We have the fasted, we have animals that were exposed to saline, animals that were exposed to EPFRs. And so saline is our vehicle inhalation control. And then we have animals that were fed a high-fat diet exposed to either saline or EPFRs. And we're looking at serum leptin, ghrelin, GLP-1, and GIP. And we saw differences or increases in leptin, ghrelin, and GLP-1 in the different cohorts. And then GIP actually increased, which is glucose-dependent insulin-trophic polypeptide, increased in the fed group, but there was no difference between EPFR exposed and non-EPFR exposed, indicating that the hunger hormones were increased, so that shouldn't be why they weren't eating more, which we were confused by and we still haven't figured out. So we said, well, okay, is their energy expenditure lower? They're eating less, hunger hormones are high, maybe they're just moving around less, okay? So to answer that question, David used the Klan system, which y'all probably are all familiar with in this department, or at least all the obesity people back there are. I'm sorry, I know this is a burden to do. But this allows real-time monitoring of food intake. This is what it looks like. This is a picture I stole from the internet. I don't think this is your system at all. Physical activity and indirect calorimetry. And so we looked at those things. And so we've got several different graphs here. Here we have the animals aggregated, okay, and plotted. And you've got different cycles. So these are the light cycles. These are the dark cycles. So when they're sleeping, when they're usually awake. And we've got the different groups. These are saline. Here we added a different control. So we didn't want to just do saline inhalation, so we actually wanted to use a particle control to make sure it wasn't a particle that was driving this response and that it was the radical species that was actually driving this response. And we didn't want to redo this experiment because it costs a lot of time and money, and our students would have killed us, right? So we used this as the control. So the particle that we used was Cabicil. That's an amorphous silica. It was the same size as the particles that we used to generate the epvers in a combustion process. And so what we did was look at VO2, so oxygen consumption rates, and this is on an aggregate basis, and this is on an individual mouse basis, with each dot being an individual mouse. And what we saw is that, indeed, the amount of oxygen consumed was actually lower in the animals that were from moms that were exposed to epvers, or who were in utero exposed to epvers. And the energy expenditure was also decreased. So here you're looking at energy expenditure. These are the control animals exposed to particle, and then these are the animals that were exposed actually to epvers in utero. And then if you look at ambulatory movement in the cage, how much are they actually moving around in that cage? You can see that the actual amount of movement, even depending on the cycle, independent of the cycle, whether it was dark or light, was actually decreased in the pups that were born to moms that were exposed to epvers. Okay, indicating that exposure to epvers in utero actually reduced energy expenditure and movement within the cage. So then we started looking at the mitochondria. So here we're taking quadricep muscles. They were 12 weeks on the high-fat diet. Quadricep muscles were, skeletal muscle was isolated, and we looked at mitochondrial copy number to see if that was changing and could explain our results. And so mitochondrial copy number was down as indicated here. So these are different mitochondrial genes, and the gene number is indicative of copy number, although there are other markers of mitochondrial copy number that we used. In addition, mitochondrial biogenesis markers. So these are genes that are responsible for mitochondrial function, upstream of mitochondrial function. Most of these markers were actually increased. So this was not the explanation for why mitochondrial function was impaired. And antioxidant defenses were up through the roof. Okay, so this is antioxidant genes, and the majority of them are extremely elevated, indicating that these pups, 12 weeks into a high-fat diet, exposed in utero, still think they're under oxidative stress. Or they're under oxidative stress because of the high-fat diet and whatever changed in their body due to that exposure in utero.
So just to be clear, the saline exposed were exposed to the high-fat diet also for this
Correct.
comparison?
Yes.
Okay.
Yes. That's a good question. So all of these, yes. I'm sorry. Yeah, that's a great question. So the saline were all exposed to high-fat diets. Right. So it's not the high-fat diet alone. It's something that occurred in utero that's being carried over and being exacerbated in the mice once they get exposed to the high-fat diet. And there didn't seem to be a diabetic comorbidity. Now, we didn't do an insulin sensitivity test on these, I don't believe. So I don't think so. But we could see that there were no real differences between the two groups in serum insulin levels or other markers like glucagon. Now, glucagon was increased in the fasted, but there was no significant difference in the fed groups. So it didn't seem that diabetes was a comorbidity. And so I'm going to bring you to the conclusions, and then I want you to think about something. So while I didn't show you all of this, we have data on all of this, and if you're interested, I can talk to you about it. But what I'm showing you here is that F for exposure in utero enhanced weight gain and predisposed to obesity. And that obesity was exacerbated on a high-fat diet, right? Now, ROS-mediated changes were observed in the mitochondria. We also observed decreased oxidative capacity and decreased energy expenditure. All of these might be related, but we don't know yet. There was inappropriate activation of PPR gamma dependent pathways, increased adipogenesis, and increased energy storage. And so what we pose, and which was going to be the background for our e-cigarette grant, was that the existence of EPFRs in particulate matter from e-cigarettes actually could lead to the toxicity associated with an e-cigarette exposure. And we were going to look at obesity. And we're going to look at that because this is what I want you to think about. So this is global PM 2.5 levels. 2 billion children lives where it exceeds international limits. So these kids are growing up exposed in utero to things containing EPFRs, and then their parents might be smoking. So, for example, smoking is more and more prevalent in China. Every time I go, I see more and more people smoking in the household. PM 2.5 levels are already through the roof in those areas. And while China used to have a very healthy diet, now when you go to China, you see Kentucky Fried Chicken almost on every corner. So now they're on a high-fat diet, and they're starting to see obesity problems. So can exposure to these things actually predict a global obesity problem that we're going to have to be dealing with in the future? Not sure, but it's kind of cool to think about. And obviously, I don't do this work anymore. I have a lab. I have a big lab. Most of the work that I showed you was done by Sridhar Jaligama, who's moved on from the lab, and David Bridges' lab, who was actually responsible for all of the obesity and adiposity and mitochondrial stuff. And the Superfund program, which helps us generate and characterize these particles, and then obviously funding from a variety of sources. And I'll be happy to answer any questions you might have. So,
following up on Rita's question, so if the in utero exposure changes something to make those pups vulnerable,